As a baby, Andrien Wald showed increasingly severe symptoms after eating and ended up in the intensive care unit because of a metabolic crisis. The diagnosis soon followed. He suffers from the rare metabolic disorder methylmalonazidurie, known as MMA for short. Thanks to continuous care from the Children's Hospital Zurich up until now, in his young adulthood, Andrien Wald has learned to manage his illness by, for example, paying particular attention to a low-protein diet. MMA affects his growth, and in serious cases, it is important to react quickly and correctly, which entails certain limitations.
Wir sind nicht wirklich viel reisen oder so, weil wenn ich entgleist werde, also dem sagt man so, dann hätte man halt müssen da ins Spital Zürich. Ja, aber wenn ich irgendwo in den USA wäre oder so, ist das ein bisschen weiter weg.
What people like Andrien Wald experience originates deep within cellular metabolism, a complex process that is disrupted in MMA. Professor Matthias Baumgartner, head of metabolic diseases at the University Children's Hospital Zurich, has been studying this rare disorder for a long time and is familiar with its various manifestations.
Die Methylmalonazidurie ist eine seltene Stoffwechselkrankheit, die im Abbau von Aminosäuren entsteht und bereits im frühen Kindesalter zu schweren metabolischen Entgleisungen führt. Im schlimmsten Fall führen diese Entgleisungen zum Tod. Ebenso können sie zu einer Metabolic Stroke, einer Schädigung des Gehirns führen, die oft im Rollstuhl endet. In weniger schlimmen Fällen wird das problemlos überstanden, die Erstmanifestation, und es kommt zu obligaten Langzeiterscheinungen, wie ganz prominent die Nierenschädigung, die über die Zeit zu einer terminalen Niereninsuffizienz führt.
To better understand the complex mechanisms of MMA, the University Children's Hospital Zurich and the ETH domain collaborated on a research project supported by cutting-edge multiomics methods offered at ETH. The project is a prime example of how patients can benefit from basic biomedical research. A key foundation for this work was a scientific treasure built over decades at the Children's Hospital.
Wir hier in Zürich sind seit über 20 Jahren und zuvor in Basel ein diagnostisches Zentrum für die Methylmalonazidurie und haben über die letzten Jahrzehnte ganz viele Zelllinien aus ganz Europa erhalten, um die Diagnose Methylmalonazidurie bei Patienten, die wie Andrien erkrankt sind, zu stellen. Früher konnte man das genetisch nicht machen. Und das hat dann dazu geführt, dass wir jetzt in unseren Biobanken große Anzahl von Zelllinien, also über 200 Zelllinien mit Methylmalonazidurie haben. Und ich hätte nie gedacht, dass wir diesen Schatz so brauchen können für Multiomics.
The project was led at the University Children's Hospital by Dr. Sean Froese and Dr. Patrick Forney. They describe how the project came about and the scientific questions guiding the analysis of the samples.
We were contacted by the ETH domain, who were looking for a large, clinically relevant cohort in order to test the applicability of their cutting-edge omics technologies. And this was a unique opportunity to partner with them in order to find out new, important things about our unique cell line collection.
We studied these cells of over 200 patients who were suspected to have MMA. To better understand it, we investigated their DNA, RNA, and proteins to get a complete picture of the disease. And our goal was to diagnose these patients and also to understand the disease better.
At ETH's Hunkelberg campus, a hub for this cutting-edge research is located. The Swiss Multiomics Center, short SMOC. The multi-omics approach enables a holistic view of complex biological systems by integrating multiple layers of molecular information. This integrative analysis reveals connections that isolated methods would miss. As coordinator of SMOC, Dr. Sandra Gutze knows the scientific and technical challenges well. She explains how the center works and its main focus areas.
The Swiss Multiomics Center was established to democratize access to molecular data in the context of personalized health. SMOC provides infrastructure and expertise to generate and to analyze data, from the genome over the proteome to the metabolome, enabling new insights into health and disease such as MMA. SMOC was officially founded in 2021 as a collaborative national platform through the support of PHR team. On the level of the patient, like in the case of MMA, multi-omics can be used to generate a molecular fingerprint, individualizing treatment decisions. With technologies becoming faster and cheaper, multi-omics will be increasingly integrated into clinical routine, making precision medicine both scalable and affordable.
Proteomics and metabolomics are two key pillars of the multi-omics approach. At SMOC, Dr. Sandra Gutze, Dr. Patrick Pedrioli, and Professor Nicola Zimboni combine their expertise to jointly analyze these layers. Patrick Pedrioli and Nicola Zamboni explain how these layers were specifically studied in the MMA project.
Proteomics is the large-scale study of proteins, the tiny workhorses that keep our cells running. In this project, we used it to see how proteins change their jobs because of disease. And this was one of the many different angles that we looked at in order to understand how genetic changes in MMA affect the body and to try and identify potential new therapies. And it was really fascinating because what we saw was how the cell rewired itself in order to try and cope with the illness.
With metabolomics and lipidomics, we measure metabolites. We measure everything that we eat, everything that our body produces or the gut microbiome produces. In the case of metabolic disease like MMA, that really helps us to understand which mutations have a functional consequence. Some of them will generate a roadblock in the MMA pathway. So by measuring metabolites, we can really discern which one are the ones that have an effect, and we can also see how the cell, the network responds to potentially bypass that. Most importantly, it tells us what is missing, because that is something that we can propose to the clinicians that could basically be supplemented in diet to potentially overcome the problem of the disease and support the normal development of a newborn.
Another part of SMOC is located at the Campus Biotech in Geneva, the Genome Center. Here, the genome is sequenced, the basis for understanding molecular processes individually and linking them with other omics data. Dr. Katrine Manick, Chief Operating Officer of the Center, offers insight into its core tasks and direction.
The Genome Center was established with the aim to promote genomic research and medicine in Switzerland, and the BHRT funds have played a key role for the Genome Center to become who we are today, especially as the genomic lead in Swiss national initiatives. We are an open and non-profit platform that provides human health-related genomic data generation, analysis and governance services to the whole Swiss research and clinical community. And what distinguishes us is that our data generation and analysis pipelines are optimized to process interoperable data for large-scale projects, but at the same time, the data and the results we deliver are also at clinical grade.
Dr. Cedric Howald, head of the DNA sequencing platform, explains how the MMA project was conducted and the challenges involved.
Quand le projet MMA a été annoncé au Genome Center, on nous l'a présenté comme un stress test, car nous avons dû générer beaucoup de données, 200 RNA, 200 DNA en un temps assez court. Et le Genome Center allait aussi être impliqué dans l'analyse des données. Ce projet nous a permis de montrer que le Genome Center était capable de produire des données de haute qualité en un temps record. Et depuis le projet MMA, nous avons développé d'autres services pour les hôpitaux, notamment le service de séquençage du génome rapide qui permet aux hôpitaux de fournir des diagnostics plus précis aux patients et d'ajuster les traitements.
To obtain meaningful insights from a biological sample, a well-coordinated process of all SMOC entities is required, from planning through analysis to evaluation. It begins with close coordination with partners, as was the case with the University Children's Hospital Zurich. Together, a joint approach was developed to analyze the cell line samples. Then, the samples were sent to the various SMOC locations for proper preparation and analysis. In Geneva, the genome of the cell samples was sequenced, meaning the entire DNA sequence was decoded. At Zurich-Hunkelberg, mass spectrometry was employed to detect and quantify thousands of signals corresponding to proteins and metabolites, all within a very short time frame of a few days. All resulting raw data were stored on highly secure servers, then categorized by data type, genomic, proteomic, and metabolomic, and, in a next step, analyzed jointly. Supported by ETH's data science expertise and continuous interdisciplinary collaboration, the team worked to extract valuable insights from these complex datasets. The effort of this joint analysis paid off. Dr. Patrick Forney and Dr. Sean Froese from the University Children's Hospital share their findings from the completed project and its impact on their research activities.
Traditional methods often fail to diagnose patients, but in our study, by analyzing thousands of data points at once, we were able to find a diagnosis for almost every single patient in the cohort. And we also found that in MMA, there seems to be a pathway that's specifically disrupted. And by targeting this pathway in the lab, we were able to successfully treat patient cells. This opens up new possibilities for future therapies. But this project also showed that an unbiased large dataset approach really is able to make new discoveries that would have not been possible using traditional methods. So I think the future for this type of research really lies in combining classical biochemistry with new big data science approaches to eventually improve the care for these patients.
This has changed the way we approach diagnosis, not just in MMA, but in all rare genetic diseases. Based on these results, we now have an ongoing study where we're trying to identify rare genetic diseases in critically ill infants, not just in Zurich, but across Switzerland. This study has also changed the way we do lab work. So we now prioritize skills and infrastructure which we would have never thought of before. And it has also opened up new therapeutic avenues which were previously unavailable.
Research like this helps make today's treatments more effective. Andrin Wald is now barely affected by his condition.
Ich bin eigentlich nicht eingeschränkt in meinem Leben groß. Ich kann mit meinen Kollegen Sachen unternehmen, kann normale Hobbys machen, einen normalen Job machen. Eben dank dem, dass ich im Kispi so gut betreut worden bin.
Rare diseases like methylmalonazidurie pose special challenges to medicine and research, but the effort is worthwhile for patients like Andrin Wald. Progress happens when different disciplines work together to better understand the unknown and create concrete solutions.